6 years ago, I entered into an underwater world on my first scuba dive. And what I noticed immediately were so many colors, tons of new marine life that I had never seen before. I was able to swim up, down, and around and explore this beautiful ecosystem. And later I decided I would definitely study this ecosystem in both college and later graduate school. Why were these ecosystems so important in the first place? Well, they are biodiversity hotspots. This means that while coral reefs only cover 1% of the ocean, they support more than 25% of marine life. The corals build a calcium carbonate skeleton that provides space for all of the fish. You can think of corals as the trees of the rainforest that provide habitat, food, and nursery space. Coral reefs are spread throughout the tropics, and so nearly every nation relies on them one way or another. Coral reefs are also the canaries of the ocean. Canaries tell miners whether the environment that they're in is healthy or not. And so a healthy coral reef supports a healthy ocean. Now, why do we care about a healthy ocean? Well, humans rely on the ocean for seafood that sustains thousands of coastal communities. We also rely on the ocean and coral reefs to protect our coasts from erosion, especially during storms. And the ocean exchanges compounds with the atmosphere, which brings us clean air and water. An interesting fact is that corals are made up of tiny little polyps, and these polyps excrete a protective mucus layer. And this mucus layer helps the coral gather nutrients and even protects it from different toxins. So you can think about it as a living boulder. You may know that corals have zooxanthellae, which are tiny algae that live within these polyps. And this algae exchanges nutrients with the corals and helps keep the coral healthy. However, as you see in this graph, each bar represents temperature around the globe. And our world is warming, both on land and on sea. And this is a sad reality because it can disrupt that relationship between the coral and its algae. Now imagine being in a bathtub that is boiling water. You would want to grab the towel and leave right away. However, corals don't have the ability to move like fish and birds do. They have to stay in their environment, and corals are even more sensitive to temperature than we are. The relationship between the algae and the coral breaks down very quickly, but luckily the coral stays alive. What you see here is white, but it's not a skeleton just yet. It's just the tissue that has expelled the algae that gives the coral its color. Once the thermal stress passes, the coral can recruit back that algae, and the coral can continue to grow its skeleton and support a healthy ecosystem. Now, scientists wondered, how have these corals been able to stay alive during bleaching? Well, new technology has shown us that there are bacteria living inside of these corals along with the algae. So this is similar to our own guts. Have you heard of probiotics? Well, we have bacteria in our guts that help us break down our food and produces vitamins that we can't produce ourselves. And so this discovery helped us understand yet another threat that harms coral reef ecosystems. Typically, the tissue of a coral is uniform in color, and underwater it even glitters in the sun. But corals experience disease, and sick corals are brought to the brink of death. Unlike bleaching, the coral tissue during disease actually breaks down, as you see in these images. There is no chance of the coral tissue growing back if there are organisms such as algae that start to take over that blank, exposed skeleton of the coral. And this is a major cause of death and breakdown of coral reefs. Scientists are still unsure about which bacteria live inside of the mucus of healthy corals that support this living ecosystem. And so I joined UGA as a PhD student to study coral reef ecosystems. I was excited for the opportunity to study the Florida Keys Reefs that I fell in love with 6 years ago. At 3 a.m., I left with excitement to see the beautiful ecosystem. And mind you, I have never been excited to wake up at 3 o'clock in the morning. As a graduate student, I put on my scuba gear and I was eager to get in the water to see that same colorful ecosystem I was expecting. As soon as I got in though, I got a knot in my stomach. I felt a little bit warm and I noticed it wasn't as colorful as I remembered it last time. In fact, it was a little bit bleak. There were no fish that I could see that I saw the last time, and I noticed that it was mainly flat, with only a few coral boulders left standing. I was sad and I was confused. Have the corals reached a point of stress that has no return and has caused this ecosystem to be so bleak? The more I swam, the more I noticed different diseases, discoloration of coral tissue loss, and in action, I was seeing these living boulders become eroded skeletons. I remembered the thriving reef that I saw 6 years ago that inspired me to go into graduate school. And in only those 6 years, have the corals gotten so stressed that they're sick and eventually dying? And if so, if the corals are stressed, are the bacteria that live in them also stressed? Sometimes the environment surprises you, as it did on that day of my first field season. And as a scientist, I needed to change my approach. Instead of studying the healthy bacterial communities that live inside of the coral and support a supposedly thriving ecosystem, I switched my question to ask, what does the bacterial community look like when a coral is sick? You could say I was at the right place at the right time in that summer of 2017, not too long ago. Over 2 days, I collected more than 100 samples of healthy and sick coral in only 2 reefs that were very close to each other. But this did allow us to control for environmental variables, such as space and also time, that allowed us to compare the bacterial communities of all of these corals at the same time. We collected the mucus from several different species of coral, 10 of them to be exact, and each of these corals independently had different diseases. These diseases included dark spots disease, wasting disease, white plague, and yellow band disease. So I started my search for corals, finding more sick corals than I did healthy corals, but I also was able to collect my samples. I used a needleless syringe to suck up that thin tissue layer outside of this living boulder that is the coral. Inside of this mucus that was in my syringe, I knew that there were thousands of bacteria represented, and each of these bacteria had different fingerprints, and that would tell us which type of bacteria they are. Once I got back into the lab, I took my syringe samples, and I applied a technique called amplicon sequencing of bacterial DNA to find out what were the fingerprints of each of these corals. And this would tell me the genus and species at times of the different bacteria in each of my coral samples. But also, it would tell me how many of these bacteria were present in each of my coral samples. We performed an analysis called beta diversity analysis. Essentially, we took the coral mucus that was collected, we took the bacteria inside of that mucus, we took our fingerprinting technique, and we came up with a representative value. This representative value was a summary value that encompassed both the types of bacteria present and their abundance. This way, we can compare and contrast the different communities in the different corals. Now this figure is a little bit complex, so bear with me. On the axes, you will notice they aren't your typical X and Y axes. Instead, these are variation axes. This means that the farther apart 2 points are, the more different the bacterial communities are, and the closer they are, the more similar they are. You will also see that each of the dots will have a different color, and these colors represent our different coral species. Now, I collected 3 of the same types of coral, and they're represented by 3 different dots of the same color. So let's take a look at one dot. It's a representative value describing the bacterial community of the coral. Now let me show you the entire figure. We see that those 3 replicates formed triangles, and that all of these triangles are intersecting. The bacterial communities of each coral, therefore, are similar to each other, and these are for all healthy corals. Now, here's a similar figure, but instead I've overlaid the diseased coral samples, which are represented by the different shapes. We have our dark spot, wasting, white plague, and yellow band diseases. And again, they're represented by different colors, which are our coral species. Our axes are still variation axes, and so they're going to tell us how similar or different the communities are from each other. Let's start with just a couple examples in this figure. We see 3 triangles that are 3 different colors. They represent the same disease, circle, white plague, and 3 different species, which are labeled here. You can see that these triangles are not intersecting, and so these communities are very different from each other. If I show you the rest of the figure, you will see that this is quite different than the figure I showed you before. There are more triangles that are more or less separated from each other. This means that the bacterial communities of each of these diseased corals are different from each other, and even when different species are infected with the same disease, they have different bacterial communities. We can summarize this data by using Tolstoy's Anna Karenina novel, where he states that all happy families are alike, but each unhappy family is unhappy in their own way. There is an Anna Karenina hypothesis for animals, and so we applied this to the coral bacterial communities to say that healthy corals are all alike, but every sick coral is sick in its own way. What does this mean? We have a new perspective to understand coral disease, which is way more complex and dynamic than we thought it would be. And this could lead us to discover new probiotics that the corals might need to stay healthy. And we want them to stay healthy because 25% of the marine life is dependent on those corals. Next, we wanted to quantify the difference between the different states of health, sick and healthy. What we did is we took the numbers of bacteria that we knew were associated with healthy corals and the numbers of bacteria that we knew were associated with diseased corals, and we counted them up. We decided to create a dysbiosis ratio. Here, I'm calling it the coral imbalance ratio. This ratio took the average abundance of the disease-associated bacteria divided by the average abundance of the healthy-associated bacteria, and by taking a log of that, we were able to create a graph where the coral imbalance ratio is on the y-axis, and we have our healthy, apparently healthy and sick mucus on the x-axis. Now, for the healthy coral mucus, we would expect that the imbalance of the microbial community is low, meaning that the bacterial community is doing a good job of working together to keep the coral healthy. However, for the sick coral, as we saw, it had a higher imbalance ratio, meaning that the bacteria were not doing a very good job of keeping the coral healthy. Now, to test this imbalance ratio, we collected samples from apparently healthy coral mucus that were part of a sick coral colony. And we tested this ratio and found that it fell right in between the healthy and sick coral mucus ratios. This shows us a transition between healthy, apparently healthy, and sick corals. So the coral disease is truly very dynamic, and now we have a quantitative way of looking at that. Now, we do have monitoring efforts that show us where disease is at, which species are affected, what types of disease, and prevalence. But this is new. This is a potential to monitor the actual health state of the coral and predict when an outbreak might be imminent. So my drive back from Florida to Georgia was truly reflective. We had caught an outbreak that happened real time, And we controlled for space and time so that we could effectively compare the different communities of the different bacteria to each other. But I wanted to do more than just monitor these bacteria. What can we do? For sure, we don't know which probiotics are appropriate for coral disease currently. But we are stewards of our planet, and there must be something that we can be doing right now. We need to do this to protect our fisheries economies and even to have clean water and air for future generations. Remember, if a healthy reef means a healthy ocean, a dying coral reef does mean a dying ocean. And since coral reefs are throughout the entire tropics, every nation is affected. And so this is a global and complex problem that we need to work together to fix. Until scientists find a cure, though, you can help scientists out. You can become a scuba diver yourself to help track the disease progression of these corals. You could vote for local and national politicians that are interested and passionate about the relationship between the environment and people. You could also educate yourself further and discuss and communicate with other people who might not know about this global problem. And lastly, you can make your own lifestyle changes, such as recycling, eating locally, taking public transportation, to make a difference in your own footprint. You have the power to act now. So let me ask you, what are you waiting for? I hope you learned from this presentation. Thank you very much.